Dear students, greetings again. We are now on lesson 9, and I would like to indicate to you that we're getting very close to the finish of this course. And what I'd like to remind you, this plant breeding framework is extremely important, and it covers two parts. The first part is what we have really concluded up to lesson 8. And it is basically covering the aspects of beginning with looking at who is the client. That's what I'm calling box 1. Who is the client? The client is the producer, the farmer, who expects to use the variety, the new variety, for his production. And you cannot create a product for a user unless you know what the user wants. And therefore, in any breeding program, even when you are looking at breeding, you want to assess the needs of the producer, who is the person to produce the new cultivar. What are his or her needs? So that you engage in the production of a new cultivar, knowing exactly which needs you are fulfilling. And that requires to do needs assessment. This needs assessment sometimes is done through the participatory rural appraisal, where views, preferences, limitations are actually collected before you get to develop any goals and a plan for breeding. And that can go on for a long time, up to a point where you are looking at combining traits that are of interest as a response to the needs assessed. And obviously, here, the starting materials are genetic resources, which you need to assemble. You put together the choice germplasm. And then, of course, it can go on between 5 to 8 years. It takes a long time. So all of those things with those boxes that have the arrows concluding with combining traits, is pre-breeding. And in this regard, we have actually covered a good part of this course in terms of those longer processes. So when everything else is ready now, we are actually walking into crop improvement, where you have the working collections that the breeder has to use. The working collections are more or less the intermediates to this process of plant breeding. So the plant breeding framework is very, very important. It's very key. It's very key. So what we are now getting into, and is the small part of this, but equally significant, is where we are now looking at how do you go about breeding. For example, one can want to still refine, develop, fine-tune the Mahangu elite variety we have, I think, It's Okashana 2 or something like that. We may want now to be getting to Okashana 3. What is it that is lacking? And given an example here is when we talk about combining traits, I mentioned before and even concluded with this issue about the multi-selection planner that you want to hit two birds with the same stone, two traits with the plant breeder stone. So we may have a trait concerned with the seed structure, and here you may want to be looking at the high grain seed yield because you've been told that the interest is you want a high yielding Okashana 3, for example. You want to combine good standability, a stand that is good, not too tall that when the wind comes, it will knock it out, and one which has minimal tillering because the tillers take a lot of the photosynthesis, the carbohydrates, and rob those that can go into a big high grain seed. You want to look at probably semi-dwarf plant because then all the photosynthesis is actually directed into the seed. So you can see that these traits are in essence contributing to the high grain seed yield, but the important aspect is that it must be white. And so can go on and look at a number of things. You can combine very many traits. Of course, it becomes very difficult. Some of those traits may already be in Okashana 2. So you begin to look at which important trait or traits that you want to further improve. And so this is what is the selection planner. Yeah, the multi-selection planner. And it focuses on about, just for example, it's focusing on what product, where, in terms of space, when, and the architecture of the plant. And then, of course, you are looking at the Easterbean pie. You may not breed all of those things at a go in the intermediate that you've derived from the pre-breeding. You may choose one particular focus, and that focus is what the product is, and then you are looking at where it will be planted, because you have certain varieties that are grown in Zambezi, for example, that really are convenient to be produced there for certain maize varieties. They may not be the same as those maize varieties that are grown in Grootfontein, for example, or are grown in at the Atlantic coast. Okay, so this is why this is also very important. And remember, you have probably looked at the characterization and or the evaluation based on botanical organs. And they are in those, the seed, the fruit, the flower, the leaf, the stem, the root, the entire plant, and so on and so forth. Then you are also may have characterized that in terms of the chronological phenologies, the developmental stages of the plant. Because when you are characterizing and evaluating a plant, you just don't do it at a seed level and stop from there. The plant keeps on moving, developing, and those various stages are the chronological order of characterization, or it may be the botanical order of characterization. You may have combined both in breeding, but of course, you may want to have those in mind as you enter the particular intermediate into a crop improvement. So I had talked about the gender methods based on breeding. And if we go back to this diagram here, you will begin to see in the lower one, where we have now the blue, is where we are now doing our work on crop improvement, where you have the GE representing the generative methods. And the generative methods are where you are crossing to generate new genetic combinations. Because as you keep on improving something at all times, you are wanting to generate new variation that you can be building progressively in the materials that you are advancing toward a final target product. So the each stands for that. Then we have the DER, DA. That is where you have the derivative methods, which involve, for example, selection to identify superior genetic combinations. And remember, in here, the very big aspect that is overarching is the breeder's equation, integrating the four essential elements of selection, which we have gone through in terms of the genetic gain that the breeder must be taken into account every time the breeder goes from one generation cycle to another one. And then you go on to the issue of biotechnology, which may have the marker-assisted selection. You may have other things like the genome-wide association studies, transgenesis, genomic selection to the generative and the derivative methods. So here again, you can see this arrow that is in pink in both ways is the evaluation, because all across the evaluation cuts across the evaluation of assembling choice germplasm, the evaluation concerning the biotechnology, concerning the derivative, concerning the generative methods. And this is where we want to go, and we'll be moving very fast. And therefore here, as we go on in the gender methods based in breeding intermediate prototypes, when you are doing the crossing, we have talked about the reproductive morphology. And if you are having a plant that has both the male and the female parts to it, hermaphrodite, you want to select or identify two of the outstanding parents of which data you have on the ones that have showed a good selection response, because you have predicted it using heritability and you have calculated the selection response and a few other things you have to look at in terms of the selection intensity and of course then the accuracy and the years per cycle. You have looked at all those. So then you have to convert one of those hermaphroditic parents into a male and the other one into a female. You do that by a process normally referred to as crossing or as emasculation. Emasculation, another word to emasculate is really castration, but we don't castrate plants, we emasculate them. When we emasculate, then you begin to cross the two superior parents. Remember, the crossing here, like I said before, is crossing good with good. It's also called mating or hybridizing or crossing. That's synonymous words. So you already have the intermediates which have the traits of interest. And remember, they may have come from centers of domestication, the origin or existing varieties that the farmers are growing but are not up to it enough. They are not refined enough. So you want to generate additional new variety by making crosses. You can also generate this by inducing a mutation, irradiating. We have the chemical mutagens that can irradiate. You can be used for irradiation. You have others that are physical. So here, in most cases, what we want to remember, the message here is normally the plant you have converted, or the parent you have converted to be a male parent, is supposed to be the donor of the trait of interest. Remember that. The male parent is supposed to be the donor of the trait of interest. That has a lot of implications in the selection methods that one may use. So then when you do, the baby that comes out of that is the F1. So one may ask, how does a breeder know that he or she has obtained an F1? Remember, F1 is not just one single seed, one single plant. You harvest several of those ones. And if you see that they are having a variation, the offspring are having a variation at F1, then you know that it is not F1, because F1 is really an intermediate. It has to have plants that all look the same. But also, a breeder can use specific dominant phenotypic markers that will manifest in F1 to confirm successful crosses that have been done. For example, this may be flower color in dry beans. If the colors all look the same, as opposed to the differences in the parents, then you know that you have an F1. So when you're doing this, the normal practice is to move the materials to F2. Most cases, this is done by selfing the F1 plants, having them met in that generation by themselves. And this is easy because then when you have the plants growing up into F1 plants, they will flower as F1 flowers. And when they flower as F1 flowers, they'll produce F1 pollens. And you let them actually self, all right? You can effect the selfing, but you can also let them self by making the pollen available among them. And then, of course, you get to the F2. And in the F2, you eliminate, you get rid of those undesirable types, those that may be poor. You move them out and you save the superior phenotypes to continue to F3, F4, and so on and so forth. This is a very common practice in selective breeding. Now, the thing I'm calling the evaluation loop of selection is this small, uh, 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 let's say, this small triangle that one can see here, this one here, this select, you cross, there is the selection, you evaluate. You cross, you evaluate, you select. After selecting, you also cross. You keep on doing this. This is a typical aspect of selective breeding. So the evaluation in the loop is what you can see as going on. And as I said before, this is where the evaluation is very important in each case of the two important activities, crossing and selection. Crossing and selection. And taking in mind that to fulfill the breeder's favorite equation, these are the things that must be taken into account. Having the selection intensity, which we now know what it is, you should be able to go back and define and understand it. Then you must accurately select, and there is a formula in doing that, and these are the various prescriptions on how to do that. And you have to use the genetic variants that you generate out of that crossing. And there are the various measures or prescriptions of how you are ensure that you are using the genetic variants carefully, wisely. And then, of course, the years per cycle. Here you want to really look at if you can go through a complete cycle of what you have done in as much less time as possible. Sometimes this can be done by having the material in the field during the growing season, and then when you have to keep on looking at what that may mean, you may actually want to irrigate or put it in another place where the rainy season is going on. What do I mean here? If you have a year per cycle, remember you are going to grow out your F1 plants. Those F1 plants that have leaves before they flower are F1 plants. Then you will have them progressing into the flower. And the flowers will now have the pollen to sort of cross, and then you have F2 seed. Are we together? Alternatively, look at this one. You got the F1 seed. That F1 seed is what you grow into the plants. Let me correct it this way, which you will now have growing into plants that will produce F2 seed. Let me do that correction, yes. That real now what you have in F2 are now the babies that came from the F1 seed that will progress into F2 seed. So you test the experimental cultivars, you compare with the existing cultivars at all times. It's very important to compare so that while advancing the generations after years per cycle, a breeder might settle on a new find based on the original objectives. Remember, the duration per cycle, like I was saying, you have the materials that were growing in the field as the plants coming from F1 seed. Then they go and they cross. And then after they cross, you get that you get the F2 seed. You can actually now go to alternative environment to complete the cycle by growing it there in the same period of time within a year, so that you have done two growing out activities within the year than waiting for another year to do just one round to complete the cycle. So you want to decrease that as much as possible. Now, I remember we are dealing with the materials crossed and then the generative one, of course, the generative method crossed. Now, you can also evaluate the materials using recombinant DNA technology, and that will be getting to plant biotechnology. A breeder can create segregating populations from homozygous parental genotypes. Then, of course, when you now have the F1, you know that all the plants in F1 will be heterozygous. And F2 will be both homozygous dominant and heterozygous, and then homozygous recessive in the ratio, not ration, in the ratio of 1 to 2 to 1. Then you extract the genetic material, you amplify the genetic segment on PCR and you run it on agarose gel. Then you can analyze the DNA bands to identify the progenies with the gene of interest. And molecular analysis is fast and not affected by environment. Then genotype by sequencing is now an in thing using the next-gen sequencers beyond PCR. Now, that is the plant biotechnology part of it. But let's get to the selection as really the major part of the triangulation. It's very important. You know, depending on the crop, selection may take several years to come up with a variety. Selection using phenotypic expression is also affected by environment, let's remember, which the plant breeder needs to take into account when designing trials. For example, let us consider that we have complex traits, yield, quality, biotic and abiotic stresses, etc. We have these major methods. We have the marker-assisted selection, right? Remember the generative method. We have the phenotypic selection. Remember the generative method. We have the genomic selection, which is actually the generative method in a way. Now, marker-assisted selection was talked about many years back, not too long ago, but it has been found not to be effective in selection of the complex traits. Phenotypic selection, the breeder's eye, is less precise and reliable despite the issue of the breeder's favorite equation and things you need to do. It's very time consuming. It has very low genetic gain per unit type, and it may not necessarily give expected results when we are looking at complex yields. Remember, most of the histamine pi traits are complex. Then the genomic selection is becoming very useful because it has high selection accuracy. It reduces the selection criterion duration. Remember, as we said before, in plant breeding, using the selective breeding or the phenotypic selection, it can take between 12 to even 20 or 30 years. That's a long time in the life of a breeder. Genomic selection is much shorter. You can create gain per unit time that is greater. You can have precise and accurate genetic gain, and it gives the expected results. Then, of course, using the genomic selection, which is changing the face of plant breeding, you can develop a crop variety much faster. All right? People say between 5 to 12 years. And then, of course, it has a lot of interest in sustaining agricultural growth when you release a new variety to feed the hungry. So we will stop at this point, and as we do so, we have completed 9A, and we should be heading to 9B, which will be sent to you as soon as these are processed. Thank you for listening, and please read your notes, because when I send out this, confirm from your notes PDF, so that when you are reading this, you are making notes on a piece of paper and confirming with the PDF notes. These notes are uploaded on Panopto for those who go to Moodle, and they're also uploaded on YouTube. So read them together. Thank you.